Good afternoon, everyone. Today, I'd like to take you through the fascinating process of mitosis and cytokinesis. These are two critical components of the cell cycle that enables life to thrive. We're going to go ahead and start by recognizing the continuum of phases that occurs during mitosis. I'm going to go ahead and share my screen. So mitosis, which can be divided into several distinct stages. Mitosis is the process by which a single cell divides into two identical daughter cells. It's crucial for growth, repair, and reproduction in living organisms. Mitosis also can be broken down into four primary phases, which are prophase, metaphase, anaphase, and telophase. During prophase, the chromatin condenses to form visible chromosomes, and we can see the formation mitotic apparatus. This apparatus includes the spindle fibers, which are critical for moving chromosomes during the cell division. The condensation of chromosomes actually begins during the G2 phase of the cell cycle. It continues as we process through the prophase. Now we're going to identify a couple of important structures, including in the process. The spindle apparatus consists of microtubules that helps segregate the chromosomes, while asters are star-shaped structures that form around the chromosomes. It's important to note that the nuclear envelope must break down to allow mitosis to proceed, easing the separation of genetic material. Next, as we move into telophase, we witness the chromosomes uncoiling, which is vital for allowing gene expression in the daughter cells. This stage marks the final phase of mitosis. Following telophase, we enter cytokinesis, the phase where the cell divides into two equal halves. Cytokinesis differs in animal as well as plant cells. In animal cells, this process involves the formation of the cleavage furrow, a belt of actin filaments that pinches the cell membrane, ultimately dividing the cell into two separate daughter cells. In contrast, the plant cells undergo cytokinesis by forming a cell plate. The cell plate develops into the vesicles that joins at the center of the dividing cell and eventually becoming the new cell wall separating the two daughter cells. In the plants, the middle lamella forms between the two new cell walls, acting as a stick layer to hold the daughter cells together. Also, it's fascinating to note that in some fungi and protists, mitosis occurs within the nucleus, and cytokinesis is the process that separates the daughter nuclei. The outcome of cytokinesis is two complete daughter cells, each genetically identical to the original cell. This remarkable process not only ensures that genetic material is accurately separated, but also distinguishes between the two methods of cytokinesis in animal and plant cell. In conclusion, understanding the difference of mitosis and cytokinesis reveals the elegance of cellular processes that sustain life, from the condensation of chromosomes and prophase to the formation of the cleavage furrow of cell plates during cytokinesis. Um, these processes are vital for growth, development, and maintenance of all living organisms.
So the next stage of mitosis is prometaphase. So after the nuclear envelope is broken down and reabsorbed by the ER, there will be that transition to the prometaphase. This is where the condensed chromosomes attach to the spindle by the kinetochores. One will attach to the centromere region of each sister chromatid. Microtubules are important in the process of prometaphase. They are captured by the kinetochores at each pair of sister chromatids. You can imagine this as if they are hooks hooking onto those and then basically ripping apart to start that or to separate. There's a large risk of catastrophe if a mistake is made during the positioning of those microtubules. Kinetochores help properly get the cell prepared to separate the sister chromatids. This leads to each of the daughter cells receiving a complete set of chromosomes. So due to that tension, that's how we'll get that separation. So there are two proposed data-based mechanisms that explain how chromosomes move to the center of the equator of the cell center. The first way is by assembling or disassembling of microtubules, and those will provide enough force for the chromosomes to move. The second way is motor proteins at the kinetochore and poles of the cell. They'll gather force by pulling on those microtubules. When chromosomes align together at the center of the cell, this will signal the next stage, known as metaphase. So it is a traditional and preparatory phase where everything is analyzed before the process proceeds. Hundreds of protein structures called kinetochores will connect to the polar microtubules, and they're arranged perfectly along the metaphase plate. So as I mentioned, you can imagine these as hooks that will hook and separate the sister chromatids from each other.
In the next part of our videos, I will be discussing a little bit more of the mitosis series. As we talk about more about mitosis, I would like to talk the first thing about metaphase. So during metaphase, this is where your chromosomes are lining up at the equator. As you can see, the metaphase plate is the imaginary equator where all of the chromosomes are going to align. The next phase I would like to talk about is the shortest but the most important phase during mitosis, which is anaphase. During anaphase, this is when those chromosomes are going to be pulled apart, creating two separate but identical chromosomes. As mentioned before, during anaphase, this is when the sister chromatids are separated at the centromere. There are two different types of chromosome movement. The first one we're going to discuss is anaphase A. During anaphase A, the newly separated chromosomes actively move towards the microtubules attached to their centromere, pulling them in the opposite direction. The next one I would like to talk about is anaphase A. And this is the mitosis stage where the spindle poles of the cells move apart, elongating the spindle fibers. The next phase I would like to discuss is the telophase stage of mitosis. During telophase, the nucleuses are reformed. and the chromosomes uncoil to allow for new gene expression. This is the final stage of the mitosis period, and during this, the new nuclear envelope surrounding the unraveling chromosomes form.